Welcome, Tech Explorers. Today, we delve into one of the most revolutionary yet controversial topics in modern science, gene editing. Technologies like CRISPR have made it possible to edit genes with unprecedented precision, offering the potential to eradicate genetic diseases, but they also raise profound ethical questions. The central debate revolves around whether gene editing should be used beyond therapeutic purposes to enhance human traits, such as intelligence and physical abilities, and the broader societal implications of creating designer babies. This discussion also explores the regulation of such technologies and the moral responsibilities that come with them. Gene editing, particularly through CRISPR-Cas9 technology, allows scientists to make precise changes to DNA sequences. CRISPR, clustered regularly interspaced short palindromic repeats, is a technology adapted from the natural defense mechanisms of bacteria against viruses. It uses a guide RNA to direct the Cas9 enzyme to a specific location in the genome, where it can cut the DNA at a precise spot. This cut enables the removal, addition, or alteration of genetic material, opening the door to a myriad of possibilities, from curing genetic disorders to preventing inherited diseases. The most significant breakthroughs in gene editing have come in the field of medicine. Scientists have already used CRISPR to correct genetic mutations in embryos that cause diseases like cystic fibrosis and muscular dystrophy. These advances promise a future where genetic diseases can be eradicated before a child is even born. However, as with any powerful technology, the potential for misuse is a major concern. The ethical debate around gene editing is complex, with arguments on both sides presenting compelling cases. On one hand, the ability to eliminate genetic diseases is undeniably beneficial. Parents could ensure that their children are born free of debilitating conditions, and the overall quality of life could be significantly improved for millions of people. However, the line between treatment and enhancement is thin. If we can edit genes to prevent diseases, what's to stop us from enhancing physical abilities, cognitive functions, or even altering physical appearance? The concept of designer babies, children whose genetic makeup has been artificially selected to ensure specific traits, has sparked heated debates. Critics argue that this could lead to a new form of eugenics, where only those who can afford these enhancements can give their children an advantage, exacerbating social inequalities. Moreover, the long-term consequences of gene editing are still unknown. While we might be able to make targeted changes to the genome, the interplay between genes and the environment is complex. A change in one gene could have unforeseen effects on other aspects of biology, potentially leading to new health issues down the line. Given the power and potential risks associated with gene editing, the role of regulation is crucial. Different countries have adopted varying approaches to the regulation of gene editing technologies. In some countries, there is a strict ban on editing human embryos, while others have more lenient policies, allowing research under specific conditions. Regulatory bodies face the challenge of balancing the promotion of scientific innovation with the protection of public health and ethical standards. The regulation of gene editing must consider the potential for unintended consequences, the need for long-term studies to understand these effects, and the importance of ensuring that this technology is accessible and beneficial to all, not just the wealthy. Moreover, the global nature of science means that international cooperation is essential. Without global agreements and standards, researchers could bypass regulations in one country by conducting their experiments in another with more lenient laws. This could lead to a race to the bottom, where ethical standards are compromised in the pursuit of scientific advancement. One of the most concerning aspects of unregulated gene editing is the potential for off-target effects, unintended changes to the genome that could have harmful consequences. These effects could manifest in unexpected ways, potentially leading to new diseases or conditions that we do not yet understand. Furthermore, the societal implications of gene editing could be profound. If gene editing becomes widespread, we could see the emergence of a new class system based on genetic enhancements. Those who can afford to enhance their children could create a genetic elite, leading to greater inequality and social division. The idea of a genetic underclass, people who are unable to afford enhancements and are therefore left behind, raises significant ethical concerns. In addition, the use of gene editing for non-medical purposes, such as enhancing physical or cognitive abilities, could lead to a loss of genetic diversity. Diversity is one of the strengths of our species, allowing us to adapt to changing environments and challenges. Reducing this diversity through widespread genetic enhancement could make us more vulnerable as a species in the long term. As we look to the future, it is clear that gene editing will continue to play a significant role in medicine and beyond. However, the ethical challenges associated with this technology cannot be ignored. The key to navigating these challenges lies in responsible innovation, ensuring that gene editing is used in ways that maximize its benefits while minimizing its risks. Public engagement is crucial in this process. The debate over gene editing should not be confined to scientists and policymakers. It is a discussion that affects all of us, and as such, the public must be involved in shaping the future of this technology. This includes ensuring that there is transparency in research, that the potential risks and benefits are clearly communicated, and that there is broad societal consensus on how gene editing should be used. Moreover, education will play a critical role in the responsible use of gene editing. As this technology becomes more accessible, it is essential that people understand its potential and its limitations. This includes not only the scientific aspects but also the ethical, legal, and social implications. By fostering a well-informed public, we can ensure that gene editing is used in ways that are ethical, equitable, and beneficial for all. The ethics of gene editing is a complex and evolving field. As technologies like CRISPR continue to advance, the potential to change the human genome, and with it, the future of our species, becomes ever more real. While the benefits of gene editing are clear, particularly in the realm of medicine, the risks and ethical challenges are equally significant. As we move forward, it is essential that we approach gene editing with caution, humility, and a deep sense of responsibility. The decisions we make today will have far-reaching consequences for future generations. By engaging in thoughtful, inclusive, and informed debates, we can ensure that the power of gene editing is harnessed for the greater good, without compromising our ethical principles or the diversity that is the hallmark of our humanity. Thank you for joining this journey of exploration, Tech Explorers. Stay curious, stay informed, and together, let's continue shaping a better, more thoughtful world. Until next time, keep exploring, Tech Explorers.